Fruit peels, long dismissed as waste, hold a powerful secret. They're rich in dietary fibers that can act as prebiotics, non-digestible food components that nourish beneficial gut bacteria and enhance gut health. The hypothesis that peel-derived fibers support a thriving, diverse microbiota is grounded in both compositional data and emerging experimental evidence, and it opens exciting possibilities for dietary strategies targeting the gut microbiome. Analytically, fruit peels are among the densest natural sources of dietary fiber. Apple peels contain approximately 2.4 grams of fiber per 100 grams, contributing a meaningful blend of insoluble cellulose and hemicellulose, along with soluble pectin, a gel-forming fiber known for slowing digestion and moderating blood glucose. Banana peels are even more fiber-rich, delivering 20 to 30% fiber on a dry weight basis, while citrus peels can exceed 30% fiber with a particularly high pectin content, qualities that position them as exceptional candidates for prebiotic applications. The fibers in fruit peels aren't inert fillers. They selectively feed commensal bacteria like Bifidobacteria and lactobacilli in the colon. These bacteria ferment pectin and other peel fibers into short-chain fatty acids, SCFAs, such as acetate, propionate, and butyrate, which collectively regulate immune function, reduce gut inflammation, and reinforce intestinal barrier integrity. Butyrate, in particular, is a primary energy source for colonocytes and is linked to reduced risk of colorectal cancer and inflammatory bowel diseases. Early in vitro fermentation studies with apple and citrus fibers show increased SCFA production and higher populations of Bifidobacteria and other butyrate-producing taxa, supporting the peel fiber microbiota connection. Peel fibers also contain unique polyphenol bound within the fiber matrix, such as hesperidin in citrus or quercetin in apple, which can reach the colon intact. These polyphenol are metabolized by gut bacteria into bioactive compounds with systemic anti-inflammatory and antioxidant properties, further linking fruit peel consumption to improved metabolic and immune health. Future research could systematically compare in vitro fermentation profiles of fibers isolated from different fruit peels, apple, citrus, banana, kiwi, and measure gas production, pH changes, SCFA levels, and shifts in microbial populations using 16S rRNA sequencing. Such comparative studies would clarify which peel fibers most effectively modulate specific bacterial groups or SCFA profiles, informing the design of targeted prebiotic supplements. Complementary clinical trials could assess the real-world impacts of adding peel powders to human diets, tracking outcomes like gut microbiota diversity, stool consistency, gut barrier markers, and systemic inflammation. This translational work is critical. While in vitro and animal studies strongly support the prebiotic potential of peel fibers, human trials will determine their efficacy and tolerability in daily diets. Another promising angle involves analyzing the differences between peel and pulp fibers in their chemical composition and fermentability. Peels are generally richer in insoluble fibers like cellulose and hemicellulose, which provide bulk and structural support for the gut, while also containing unique soluble fibers, such as the high-methoxyl pectin found in citrus peels, that swell and form gels, promoting satiety and feeding gut microbes. These differences could be leveraged to create custom fiber blends tailored to specific health outcomes, such as reducing constipation, managing blood glucose, or enhancing microbiome diversity. Preliminary evidence already shows functional outcomes. Orange peel fiber has been found to increase mucus secretion in the colon, strengthening the protective barrier that prevents pathogenic invasion and inflammation, a key defense mechanism in gut health. This suggests fruit peel fibers not only nourish beneficial bacteria, but also directly support the gut's structural defenses. In terms of application, fruit peel powders could be developed into standalone prebiotic supplements or incorporated into everyday foods, granolas, snack bars, baked goods, or smoothies, to create fiber-enriched products with clinically relevant gut health benefits. Importantly, valorizing fruit peels as prebiotic ingredients reduces food waste, supports sustainable food systems, and provides cost-effective, plant-based fibers with clear health advantages. Altogether, compelling evidence supports the hypothesis that soluble and insoluble fibers in fruit peels serve as prebiotic substrates, promoting beneficial gut microbiota and contributing to overall digestive and systemic health. By exploring in vitro fermentation profiles, conducting clinical trials, and developing peel-based fiber products, we can unlock the full potential of these abundant, overlooked nutritional resources to improve human health while advancing sustainability in food production.